And then the third piece that came up, and that will lead us to our next panel, is another theme, Chris, that you brought up at the very beginning of that panel, which was reinvention, that we have to constantly reinvent ourselves in our industry. And I use that as a prelude into this panel because it makes me think of the USAIC, where we have reinvented ourselves in the following way. When I started with this organization maybe 12 or 15 years ago, it was almost entirely male. It was almost entirely male, and every male would wear a blue suit and a tie. And the quality of the conversations was nothing like the diversity of conversation that we have today. And so it gives me great pleasure to introduce the next panel, in part because all of the panelists are non-male. The moderator is a male. His name is Steve Uden. This is a panel on rare and neurological diseases. Steve is co-founder and CEO of Rallybio, with over 25 years of experience in R&D leadership roles in global biopharmaceutical and biotech companies. He was the former head of research at Alexion Pharmaceuticals, where he led collaborations and expanded research into small molecules, RNA-based therapies, and protein engineering. He also had significant roles at Wyeth, who can remember Wyeth, Novartis, Oncology, and Pfizer, which bought Wyeth, across the UK, Japan, and the United States. Steve was trained at the University of London St. Thomas's Hospital Medical School. He practiced clinical and academic medicine within the UK's National Health Services and at the University of Manchester. Steve, please join me in welcoming Steve to the panel.
Thanks, Andy. I'm presuming, Karun, I'm going to have to stand because I think...
Everybody has to do the panel, all the panelists.
Okay, well, panel, let's all come and join me. Come and join me. We've got... let's all find our seats.
I think my seats are safe. We've got assigned seats.
We've got assigned seats. We've got Daphne here, Fiona there. We've got one seat missing.
One more. There's 6 of
Just
us.
as I thought.
6.
You see? Oh, well, I'll tell you what, I'll stand. How about that? So what I'll do is, while we sort out the chairs, I'll just introduce myself briefly. One of the great things about this particular meeting is a bit like you're kind of coming home. Always, a cast of characters. Andy chairs it. There's Karun chasing us on the time. Bill opens the sessions, etc. If you're all looking around for Martin Mackay, who's a feature here, he's not here. He's the chairman of our company. That's why I got asked to stand in for him. And he's currently he's on the board at Novo, and unfortunately that clashed. So I'm a poor substitute. Anyway, to get this panel started on neurodegeneration and rare disease, what I'm hoping is that this panel's going to be a bit like when I read The Economist. Okay? So I open The Economist, and you go through if you like The Economist, I'm a great fan of it. You read the leaders, and it's like, oh God, really? And then you switch to the science and technology section at the back, and it's great. So we're going to get going on what we're all really here for, which is all around science and innovation. Now, it's an interesting mix, I think, neurodegeneration and rare disease, but I think it's a really good mix. It's really good, I'm hoping, going to talk about our industry. I'm old enough that I was working in Sandwich when that was part of Pfizer, and I was leading a sort of phase 2 group, and we were working on something called UK 92480. So people might know where this story is going. We were looking at it in heart failure and hypertension. And Neville Jackson, who was a prof of cardiology, worked with me. He came to me and said, those people in the experimental medicine group, and you're looking at erectile dysfunction. I said, take no notice of them. They're all mad. And of course, 92480 was sildenafil, which became Viagra. But the innovation continued, and it became Revatio for pulmonary hypertension as well. So I'm hoping we're going to build these links. So I'm joined on the panel. Make sure I get all the names. Yeah, we've got Zandy at the end, Zandy Forbes, who is the president and CEO of Meira Therapeutics. We've got Nancy Andrews, the executive vice president, CSO at Boston Children's Hospital. We've got Fiona Elwood, who's VP of Neurodegeneration Strategy seamless end-to-end role, Johnson & Johnson. We've got Daphne Zohar, who's the founder and CEO of Seaport Therapeutics. And sitting next to me is Priya Singhal, who's head of EVP and head of development at Biogen. So with that, we're going to get started. And I thought a good place. So let's all be optimistic, everybody, and let's think about what are the great breakthroughs and exciting advances that are happening in our field, and how you think they might be able to be translated into, so if you're doing rare disease, maybe into neurodegeneration or vice versa. And because you and I are outnumbered, Nancy, I'm going to start with you to get going on rare disease, the big excitement things, and how you might think they may translate into advances in neurodegeneration, for example.
Yes, am I on? I am. So at Boston Children's Hospital, we have actually a very big effort in rare diseases. Most of them present in childhood, at least most of the monogenic rare diseases. And the way we're looking at it is by what we call cohorts, looking at phenotypes, going deep into that phenotype, finding rare variants that are causing diseases, often not previously known, and making a diagnosis. And that's great. We're getting really good at making the diagnoses, but I think where the future is, where the excitement is going from that diagnosis to treatments. And a couple comments about that. First, I think our cohorts, phenotype cohorts perspective allows us to learn more about the biology than we might from a different effort we have, which is just to diagnose undiagnosed patients. But we're very interested in moving from knowing what a variant and the biology tell us to being able to find a therapy. I think gene therapy and antisense oligonucleotides have been ways to do this. I think we need the next generation. I don't think those are going to be ultimately the answers that we're really looking for. We need next generation approaches that ideally will, in relatively short time, be plug and play, so we'll be able to identify a variant and move to a treatment. And ideally, these plug and play treatments yet to be invented will also serve not only the global north, but also the global south. So I think there's a lot of excitement there. A big problem standing in our way is a business model to support treating patients with rare and ultra-rare diseases.
And I think we'll definitely get onto that as we move forward. Now, you can see Zandy is sitting on the edge of her seat because we did a really good prep session, and she told us about something. If we could have canned the prep session, I was saying we could have all gone and had coffee while you just listened to the preparatory discussion. But Zandy, tell us about some of the interesting stuff that Meira have been doing recently.
So we are a genetic medicines company that treats both rare and very common diseases, and recently have had a lot of success with an extremely rare eye disease where children are born blind. We treated 11 children. All of them can now see, actually see, read, etc. And this is a story of regulators coming to us to want to approve something in this environment which really works, to Chris's point. If you have a big effect, we've treated 11 patients, they came to us and want approval. So that's in the rare area. I would say the area that applies to all of us is something very exciting that we've done with an AI company. And we've heard a bit about AI and how it can help in drug discovery in many areas. But what we did is we formed a joint venture around our neuro programs, a Parkinson's treatment which circumvents the need for dopamine by rewiring the brain. Now, we had a positive phase 2 study with UPDRS, successful, and hints on brain scans that we were doing something to the brain. So we formed a joint venture with this company that came out of the Wellcome and UCL. It's been working for 10 years to address the issue of clinical data. Not trial design, not drug discovery, but how do you see real effects, robust statistical effects in the heterogeneity of clinical data. If you can do that, you don't have to do 2 phase 3s. If you can show with 20 patients in each arm, a really, really real effect, you've done it. You can take it to the FDA and your development is curtailed. So we gave our data, 20 patients, positive study, to these guys. They came back and they showed us inside the brain. They showed us actually, and this is statistically, a rewiring of the brain, an improvement in the nigra that was disease modifying, and though symptomatically this improved motor symptoms, we changed cognition and mood. So this is AI being used to show reality. Our current clinical trials are so noisy, measures heterogeneity, that we have to do large studies multiple times to convince ourselves and the FDA things work. Now we're going to be in a position, we did a JV with this company, to use it across the board for clinical data. Not to design trials, but to look at the truth of what's happening. Sometimes things may not work, but we'll know they don't work, so we don't continue. So we de-risk and we see the truth a lot sooner, and it's super exciting.
So if you want to see scientific enthusiasm, there it is. So Fiona, you and I were chatting just before, in the clinical trial session, and you were telling about some of the breakthroughs in your space and how, some of the links. And I believe that your group has just had its first approval. So perhaps you can share that with us.
That's right. We were very excited that Imavi, nipocalimab, was just approved last week. But maybe the place where I think about that bridge from rare to neuro, to neurodegeneration, and to prevalent diseases is really in that precision approach. Because I think precision started in rare diseases with that genetic basis of disease. And now I'm really interested in how we can take that precision, that deeper understanding of the stage and subtype of diseases like Alzheimer's and Parkinson's, to really target our molecules to the right patient at the right time, to measure the, as you were saying, to have that greatest opportunity to measure the benefit of the therapies. And honestly, Alzheimer's is a place where we've seen massive change over the last 10 years. 10 years ago, when we recruited patients to clinical trials, 20% of them didn't even have Alzheimer's, because we were recruiting people based on clinical symptoms. Then we went through the PET imaging and the ability to image and see inside the brain what the state of the pathology was, the targets that we were going after. Now we can stage patients with a blood test, not just diagnose patients, but with our most recent conference, even see the stage or the progression of that pathology through the brain based on a signature of fragments that you can measure in the blood. That's amazing, because that means that you're enrolling the people who are most likely to benefit from your therapy. You can identify them even before they start to develop symptoms. So in neurodegeneration, where going early, where we want to prevent cognitive decline, is really important. Having a high throughput way of identifying, recruiting, enrolling those patients is really, really important. So that's a place where I'm particularly excited about today. This isn't even the future. This is where we are today.
Yeah, it's great. And nipocalimab actually is another great example, as you and I were discussing, that although it's an FcRn inhibitor, and your first approval is in neurodegeneration, but as we discussed, there's work going on with the maternal fetal medicine, looking at mothers who are alloimmunized in these very rare, what are fortunately very rare conditions these days. So again, cross-fertilization across a...
Absolutely, and a super exciting pathway, the being able to target autoantibody or alloantibody diseases, which we're learning more about across multiple different diseases, indications, from rheumatology, maternal fetal, even to neurology indications.
Now, Daphne, you're somebody who's really been committed to CNS and neurodegeneration and neuropharmacology altogether. It'd be great to hear your thoughts about, what do you see as the big advances sort of in terms of therapeutics or any aspect of the work that we do?
Right, so I'll say a little bit about what I'm personally excited about, what we're working on, and then I'll talk about some things that I think are intriguing more broadly
Sure.
for the field and perhaps beyond. So one of the areas that we've been focused on as a strategy is looking at medicines that have previously shown clinical efficacy but had an issue that held them back from really making an impact on patients. And we pursued that strategy at my previous company, PureTech, where we founded Karuna and invented Cobenfy, or was originally called CAR-XT. So this idea that you could take a drug that had previously shown efficacy and then do something with today's technology, today's insights, to enable that medicine to really have an impact is one that I'm really excited about. It's one that we're pursuing at Seaport. Now, we talked about this progression from rare to neuro, and then we're focused on neuropsychiatry, so it's kind of like the opposite of rare. The prevalence is really rivaled only by obesity and hypertension. So these are really big diseases. We have this issue of heterogeneity and the ability to really understand which drugs are going to work for which patient. So if you look at an all-comer MDD study, 50% of patients aren't going to respond to SSRIs. So how do we get a better understanding of which patients are likely to respond to drug and to placebo? And here's where I think there's some interesting work happening. We're not in the area yet in neuropsychiatry of blood-based biomarkers, but we're really, I think, making progress in the area of functional imaging-based biomarkers, so EEG-based biomarkers, fMRI, et cetera. And there's some really intriguing work happening in that area in looking at very large data sets to identify patients that respond to certain drugs and also patients that respond to placebo. And I think that's really intriguing, especially if it could be used beyond neuropsychiatry.
Yeah, definitely. Great. And Priya, if we're going to think of a company that's committed to neurodegeneration and has touched on rare disease as well, I can't think of any better company than Biogen. So it'd be great to hear your thoughts about, big advances and things that you're excited about.
Thank you. I'm excited to be here with all of you.
We all are, I think.
And, I think there's several themes that have emerged, but before we talk about the themes, for drug development, which I think has single-handedly changed human survival for the last century, I want to say that there has to be a tipping point. We've seen that tipping point for Alzheimer's disease. To Fiona's point, I think it was about amyloid PET imaging that changed the story, making sure we had the right patients in the study to be able to test a therapeutic. And I think we're seeing a lot of advantages and advances now. And I think the cross between neurodegeneration and rare is quite, it's very interesting, because we have actually seen the importance of two aspects. I could touch on many, but I'll touch on two and I'll give you some examples. Number one is that it's an ecosystem and it's a team sport. We cannot do this on our own. We need the community, we need the academics, we need other sponsors, and we need to be able to come up with tools. And I think Zandy touched on it, because we simply cannot afford to do these large trials. So I will tell you the tools that we think are going to change the world in many ways and are already on the way to changing the world. One is biomarkers, which has been talked about a lot. And I'll give you 3 examples, which are personal examples at Biogen. Number one, we had SOD1 ALS Qalsody, which is now approved. But we failed the main study, the pivotal study, because we did not have a good sense of natural history. And patients and natural history didn't behave in the same way. Luckily for us, we had a tool called neurofilament, and we were able to anchor advances that patients were making in terms of their progress based on neurofilament. None of these we could have done on our own. Fast forward to Skyclarys, where we merged with, we acquired Reata Pharmaceuticals. Skyclarys is for Friedreich's ataxia, neurodegeneration, but also rare. And the reason that it got approved was because FARA, the Research Alliance Group, had done a 20-year natural history study, and the MOXIE pivotal trial long-term extension was able to show a clear differentiation. Number 3, fast forward even more to very recently, we announced our collaboration with Stoke Pharmaceuticals for zorovanersen, which is again a rare, very important genetic encephalopathy, Dravet syndrome. And here, it's even better because they did the natural history studies and they were able to incorporate elements for biomarkers, tests, and measures in the pivotal trial design. So we're still to do the pivotal trial, but we feel good about what the design is set up to show. And so I think that the tools are critical because it's very easy to say that, hey, these are financially constrained times, we need to fail early. Well, how do you fail early? You need to have confidence that you've either tested the hypothesis and it doesn't work, or that you have a signal and you've identified it and it is worth investing in.
Isn't this great? So all this stuff, I used to come to these sorts of conferences 10, 15 years ago, and things like biomarkers and artificial intelligence were thrown out there. And here we are actually hearing it sort of come home, and genetics, of course, coming home to be successful. Now let's just change track a little bit, and we're going to talk about the next set of challenges that we're going to be facing on the sort of scientific and translational front. And I know two topics came up when we were on the call. One was the sort of, whilst we've been making great progress in Alzheimer's disease, what about Parkinson's disease? And that came up several times. But then, of course, always this whole issue of the business model with this burgeoning of innovation. How do we get the financing needed to get the job done? So let's start with Parkinson's disease. And I think it was you, Daphne, wasn't it? Or was it Zandy was on? No, somebody was on a...
Maybe I
Fiona.
can just
Fiona,
jump in
sorry.
here.
Remember, I was doing this on a telephone conference, so you're trying to put the face to the voice all the time.
So we talked about biomarkers in
Yes.
Alzheimer's and how these amazing public-private partnerships, natural history studies have really enabled us to get to this point we are today where we can have a blood test to identify patients even before they develop the disease, develop the cognitive symptoms. So that has really transformed what we can do in that space. In Parkinson's, we've got some of those elements in place. We've got some great natural history studies, public-private partnerships, Michael J. Fox Foundation, PPMI, for example. We just had the first, last year, CSF biomarker measurement, where we can measure the presence of aggregated alpha-synuclein in a patient's brain. We're just having the first proposals for a biological basis of disease and staging of disease that can help define the patient population from a biological perspective that we want to go after. But the gap that we have, and maybe Zandy talked about this a little before, is how we run those clinical trials. If we need to advance these biomarkers, there's still more that needs to be done. But then once we recruit those patients, how are we going to measure that rate of progression in patients? And actually, just at our last conference in Vienna a couple of weeks ago, we saw two trials, one from Roche, one from UCB and Novartis, where they really came up against this challenge in Parkinson's, that when you've got a symptomatic treatment, patients can be given increasing levels of the symptomatic treatment, and any impact on disease progression can get masked. And so I think that is really the area where the tools can come in, or different ways of analyzing patients to really try and identify the rate of progression of disease and an opportunity maybe from other disease areas to really potentially impact in the Parkinson's space.
Zandy, have you got any thoughts about this?
Yeah, I've got lots of thoughts,
Oh.
but I'm going to keep them to a minimum to respect Corinne's timeline.
Oh, yeah,
Okay?
be careful. Yeah.
I think when we think about biomarkers, we're thinking about measures that predict who will respond and who will not respond. So reducing the heterogeneity in a subsequent clinical trial, which we're starting to get more tools to do. But we can also think about if we can see inside the brain, right? We remove the heterogeneity, we can watch or model what the progression would be. What we can actually do is have proximal endpoints to the disease. So now in our hands, in Parkinson's disease, we can look inside the brain and the endpoint is rewiring, really statistical, really, really robust. So we have got from symptomatic in what we have to show clinical meaningfulness, but we now have an objective measure that reflects the mechanism of the treatment, which is another thing that's really required for approval. And they're often disparate, right? Clinical meaningfulness is some PRO score, but objective measure is how much of this or that do you have present. Now, what's interesting about what we've done is we didn't affect dopamine, right? Nothing to do with dopamine. We're not going after synuclein, the cause of the disease. However, what we've done is change one locus of a neurotransmitter that rewires the brain in a way that now produces GABA, which protects the brain, compared to glutamate that kills it. So there are many ways we're now understanding of disease modifying, right? In Alzheimer's, are there circuits that we can hijack to increase your cognitive flexibility for the next 10 years? That's disease modifying, but it isn't changing the cause of the disease. And this circuitry and being able to change circuitry and identify, using this technology, what nodes in the circuit to treat is something completely new, to me anyway, until this year, and very powerful, a very different way of thinking about things. Circuitry and proximal endpoints.
I can see Fiona on the edge of her seat there. So come on, Fiona, off you go.
And actually, Daphne, you
get
brought
Daphne
this up
to jump in as
just
well, yeah.
before. The news flash is we have one brain. We don't have a neurodegeneration or a neurology brain and a psychiatry brain. All of our training and the way we organize our sort of, training systems, we separate neurology from psychiatry, but it's one brain. And whether we're looking at the biological basis of disease, essentially the cell biology of neurodegeneration, or we're looking at the synapses, the circuits, and how our brain connects, it is one disease. And I love this convergence of the neurology and the psychiatry, and maybe where we're going to learn from each other and where we're going to go further by taking the strengths of both.
This is not bad for a challenges section, is it? It's doing really well. Daphne.
Do you mind if I say one thing?
Of course you can,
Fiona?
yeah.
Because it's so important, right? We do have one brain, and the heterogeneity that we see amongst brains is people with Alzheimer's and depression, bipolar people with Parkinson's. So the data that this particular AI was based on actually started with all the longitudinal data from Queen's Square. 20 years, every neuropsych and every neurodegenerative patient that had been through that hospital, real world and clinical trials. And that is what they test our data against. So it's not Parkinson's patients, it's brains, exactly like Fiona said. Super important.
Well, let's get Daphne to jump in because you're looking at these big diseases. So, any thoughts about some of the challenges and whether any of these technologies will help? And then we'll get to you, Nancy, on the and then Priya, you could bring it all together into one big
Yeah, so I love the direction that this is going in terms of the ability to translate from one area to another, and I think that's going to be really important. In terms of the challenges, I think the biggest challenges that we've had in the area specifically of neuropsychiatry, but also more broadly, is the lack of translation from preclinical to clinical efficacy. And that's really why we're focused on starting with drugs that already have clinical efficacy. The second is that the drugs that we have for depression and anxiety, these are drugs that are old drugs. They have modest efficacy. A great percentage of patients don't respond. They take weeks to work. But even with that said, if you look at the drugs that have been approved for depression, for example, every one of them has been a commercial success. So that's pretty astounding to think about.
Yeah.
That speaks to the unmet need, the breadth of the population that could be impacted. But we're really in early days in understanding what, is this one disease or are these multiple diseases? And really, I think, following in the steps of what's happened in areas like oncology. So I think the biggest challenge, though, for neuropsychiatry in particular is running clinical studies, and there's a lot of learnings that can be shared. So, there's a lot of things that I think those that have done this have sort of developed scar tissue around, and that is very high placebo responses. And there's ways to manage, for example, scrutiny of enrollment, limiting the number of arms, limiting the number of sites, limiting in particular the number of assessments. And I think what's really important, Chris earlier talked about shared resources. So I really believe in sharing information, sharing data. We talked a little bit about that. So there's been some really great work done in studies like EMBARK and STAR-D and CanBind, where you get shared data that is then put out into the community where it can be analyzed with AI and with other tools that we have available. And then I think that taking some of these learnings and sharing them, for example, one thing we did as a company, which I think might be helpful, is we did a meta-analysis of all the different clinical studies in depression, and we shared the learnings about what contributed to higher placebo response. So those are some of the challenges and how we're dealing with those challenges. But I'm really excited about what I'm hearing from others here on the panel.
Priya, will you just make a quick comment about it, and then I promise we'll get to you, Nancy.
Yes,
Yeah.
I wanted to pick up on the Parkinson's disease.
Oh, right, yeah.
Because it really has been a graveyard of trials. So there's a lot of investment, but there's really no disease-modifying therapies. And one thought that we are thinking about very deeply is the use of AI. So prognostic covariates. And I think it is an area that's very rich with data. So how do you bring it together to identify real natural history and to address the heterogeneity of disease and identify signals for therapeutics? We're doing that for Alzheimer's disease, which I think is probably a decade ahead, but I think there's a lot of translation possible for Parkinson's disease. So I'm excited about that being the next frontier.
This is great. This actually is a good segue to Nancy, because if we're going to talk about translation, right at the beginning of the funnel are people like Nancy. So what are your thoughts on this and some of your challenges?
Yeah, thank you. So first, I realize I didn't answer your initial question, which was how does neurodegenerative diseases relate to what we do in thinking about rare diseases. And there is a very close connection. Just a few thoughts building on what I've been hearing from my panel colleagues. First, I think that neurodegenerative diseases are a disproportionately large piece of what we see when we're looking at rare diseases in children. And I hadn't thought about it until just now, but there may also be untapped opportunities to think about the whole brain, the neuro disease, psychiatric disease, how they blend together and how they don't in using rare disorders in children as a means to discover new potential targets. For us, a big problem with rare diseases is now that we're really good at making the genetic diagnosis, not always, but a lot more than we used to, trying to deal with the situation where there may be one child in the world or 3 children in the world who have a particular variant that causes a really bad disease. And we think we know how to approach it or begin to approach it therapeutically, but we know that there is no market at the end. And how do we deal with that problem that we can only get so far in academics, and then somebody else needs to take it over and take it the rest of the way? And I have 3 thoughts about how we can begin to approach this, and these are not very mature thoughts, but one is bundling, and there are ways that we might be able to bundle rare diseases with similar treatments or with similar pathways and make it a bigger market somehow that way. A second thought is drawing on what you were, I think, what Zandy was saying about modifiers, or maybe it was Fiona. If we don't go after the gene itself, but go after something that will modify this rare disease, then that modifier might have more applications and might have a bigger market. And then finally, and this is obvious when you see it, but not always up front, a lot of the rare diseases that we see in children have milder equivalents in adults. No surprise, genes can get many kinds of variants. In kids, they might be variants that cause a severe phenotype that you pick up early. In adults, you might not even be aware that there's a phenotype, but it still might be something that would be worth treating. And so to the extent that we can predict what an adult variant or a milder variant would look like in an adult, that could increase the market as well for rare diseases. So just a few thoughts. And I welcome anybody here who wants to talk about it further to try to think about
this.
This
is great. We've got everything from Nancy worrying about 1 or 2 patients, 4 or 5, and then Daphne, like, we've got this huge public health problem to solve. We could just carry on talking amongst ourselves, but unfortunately, we have to take the surprise questions from the audience. And I believe there's a surprise question from Sam Elwood on table 1. For us. Here he is. I was rather hoping Sam wasn't here. We could just carry on chatting amongst ourselves.
Hi, thank you for a great conversation. Thinking of new targets and bridging from ultra-rare to more prevalent indications, how do you find better targets for more disease-modifying treatments? That's what I want to hear about.
Oh, great. Who'd like to start? Probably Fiona. Go on, kick off, then Zandy. I'm sure has got some ideas. Yes, Fiona.
I think one thing that I'm particularly excited about that we're have invested in along with the Gates Foundation is the Global Neurodegeneration Proteomics Consortia. And actually this comes from, Zandy's point about looking across multiple different diseases. And, whilst you were talking about brain imaging and functional connectivity, this is a proteomics resource that looks across multiple different neurodegenerative diseases, 40,000 patients, discovery proteomics, 7K analytes per patient. That's going to be made public in July. And that's an opportunity to look across diseases. So to look across diseases for pathways that are dysregulated, not just in Alzheimer's or Parkinson's, but in potentially rare diseases or psychiatric diseases as well. And an opportunity to really look for targets that go beyond our clinical definition of disease and really that are grounded in common biological pathways that can be dysregulated, but depending on many other functions, factors, can be expressed or observed differently in different populations.
Very great. Zandy, you want to add? I'm cognizant of time, so I think everyone's going to rather stay and listen to us than go and have coffee. That's poor network. That's my belief. But were you going to add anything to that? Or?
I could add something, which is when we think of target, we think of a molecular target or a drug target. However, when we're dealing with neuropsych and, in fact, neurodegeneration, we can have circuitry targets. And one of the great powers of this new technology to look inside the brain with AI allows you, particularly in neuropsych, to identify very specific loci in the brain that you can locally change to change the circuitry. So it's a new way of thinking about targets that we're now able to address.
I was just going to add that, our collaborations, and I think it's been mentioned, but with the UK Biobank, with Our Future Health, that's our way of looking and identifying targets. But I want to go one step further because it's not adequate to just identify the target. The question is, how much do you need to modulate it? How are you going to measure it? And an example I want to share is about tau in the brain. And one of the early questions we had many years ago was, how much do you need to reduce it? And how would you measure it? And then, we looked at the UK Biobank and we really were able to show that people with a natural loss of function or a haploinsufficiency of tau were walking around. So we knew that 50% was compatible with life. And that was an important anchor that then we built our program on.
That's great. So we're going to have one more surprise question. And then I think we're going to have to declare defeat. We can carry on talking amongst ourselves, but... sorry?
Declaring victory.
Declaring victory, sorry. No, defeat, because we weren't able to keep the conversation going. Oh. Yeah. Look, this team, we could be here. This is like, I come from a family of women. I'm the only boy in the family. So this is just like me being at home. So it's great. We just carry on all. So Chris Benko, I believe, on table 13 has got a surprise question for us. Nope. Oh, I'm looking in the wrong direction.
Question.
Sorry, Chris.
So, no worries. So it would appear the US FDA is signaling a continued move toward accepting surrogate endpoints and less emphasis on necessarily having early stage placebo-controlled studies for rare diseases. Many of us would favor that. However, those changes don't necessarily align with market access and reimbursement in the US, nor do they align with the direction of the EMA. So I'm curious as to whether or not you view these changes as being significant in terms of your optimism and the way you might change your investments, given that I know that all of you have a goal of serving all of the rare disease patients around the world. And so does that divergence give you a window of opportunity, or do you really depend on everybody else catching up for that to truly make a meaningful change to how you invest and develop these medicines?
I think we'll start with Nancy, and I'll see if Daphne can come up with anything. When you're here, you're trying to deal with these huge... Is there any links? But Nancy, why don't you start?
Well, the first thing that comes to mind is when the FDA is asking for placebo-controlled trials on vaccines, as they seem to be, I wonder how long anything they say is really going to last. I think that, as Zandy was talking about, there are situations where it can be pretty obvious that you've got what you need, and perhaps that would be accepted in other parts of the world as well. Beyond that, I don't really have a whole lot to add because I'm not in this space other than on boards. But at the moment, I think the FDA has become a work in progress.
Sorry, Daphne.
Yeah, I mean, I was intrigued by Marty Makari's comments recently about rare disease and the potential for approving a drug without a randomized control study just based on a plausible mechanism in the area of rare disease, and then tracking, following that by looking at medical record data. So that was intriguing. How would it work? You pointed out all the challenges that would come into play. I also think it's very promising to hear that they might be working towards one randomized control study instead of two for approval. And so for us, that would be huge. I mean, I think it would be a great thing. I agree that we don't know, like the administration, there's a lot of movement, but I think Makari's viewed as being somebody that the industry can work with and somebody reasonable. He's not sort of in the crazy Maha camp. So, I'm somewhat hopeful, and I think the industry also reacted, like the XBI, IBB, reacted positively to some of those statements.
So Priya, and then we'll do Zandy, and then we're going to have to, and maybe Fiona, we're going to have to go and have coffee, otherwise we'll be in trouble with Priya.
I want to say that we have a sort of an example here with Qalsody and Tofersen, where we had a surrogate endpoint, and we came up with it in the trial. It was a placebo-controlled trial, but it was a trial that failed on its primary endpoint. We were able to use the surrogate endpoint, not only in the US, but also in Europe and Japan and other regions. So I think we have seen that we can break the barrier. The devil, which is in the FDA, I think, is in the details. I think the details are really important on surrogacy, validity of the surrogacy, and I think payers are a whole separate conversation. So I think more needs to be done from a health economics perspective to be able to bridge that gap, which is the ultimate sort of, can you reinvest into R&D? So I think it's a big one.
Oh, right. So it looks like we're going to have to declare defeat, as I said. Well, it's been a great panel. I've really enjoyed myself. Let them all go and have coffee, and we can keep talking amongst ourselves. So thank you, everybody, for your attention. Hope that you enjoyed this.